Welcome to today's lesson on inherited disorders, where we will explore the genetic basis of certain diseases and the implications of embryo screening. In our lesson today, you'll understand how some diseases, like cystic fibrosis and polydactyly, are inherited through specific genetic mechanisms. We will then discuss the economic, social, and ethical issues surrounding embryo screenings. So far, you have learned about the key concepts of genetic inheritance. This foundational knowledge sets the stage for us to introduce how probability and ratios are used to predict genetic outcomes, and how complex inheritance patterns can be understood. Understanding these concepts helps explain how simple and complex traits are inherited. This insight is essential for fields ranging from medical genetics to evolutionary biology. Inherited disorders are medical conditions that are passed from parents to offspring through their genes. These disorders occur because of specific alleles, which are different forms of the same gene. Some well-known inherited disorders include cystic fibrosis and polydactyly. Let's explore these in more detail. Cystic fibrosis is caused by a recessive allele. We will represent this as a lowercase f. This means an individual must inherit 2 copies of the defective allele, one from each parent, to exhibit symptoms of the disorder. Cystic fibrosis results in the production of thick, sticky mucus, primarily affecting the respiratory and digestive systems. This mucus can block air passages in the lungs and ducts in the pancreas, leading to significant health issues and reduced life expectancy. Let's discuss how cystic fibrosis is inherited. Carriers have 1 copy of the recessive allele, denoted by capital F lowercase f, and do not show symptoms. If both parents are carriers, there is a 25% chance their child will have cystic fibrosis. This occurs when the child's genotype is lowercase of lowercase f. Using a Punnett square, we can see that the possible genetic combinations are 25% chance of capital F, which results in the individual being healthy, a 50% chance of capital F lowercase f, which represents an individual being a carrier and having no symptoms. And then finally, there is a 25% chance of the individual having a genotype with both alleles being lowercase f. In this case, the individual will be affected by cystic fibrosis. Let's now discuss polydactyly in more detail. Polydactyly is caused by a dominant allele, represented as a capital D. This means only 1 copy of the allele is needed for an individual to have the disorder. The disorder results in extra fingers or toes. Unlike cystic fibrosis, polydactyly usually does not cause other health complications. Only 1 parent needs to have the dominant allele, D, for their child to potentially inherit polydactyly. If 1 parent carries the allele, with genotype capital D lowercase d, and the other does not, there is a 50% chance their child will inherit polydactyly, also with genotype capital D lowercase d. The possible genetic combinations can be seen with a Punnett square as shown. In this case, there is a 50% chance the individual has genotype capital D lowercase d. In this case, they will be affected by polydactyly. There is also a 50% chance the genotype will be lowercase d. In this case, the individual will be unaffected. Embryo screening involves analysing the genes of embryos to detect inherited disorders. This is commonly done during in vitro fertilization, IVF. Let's discuss the IVF process. In step 1, embryos are fertilized in a lab. In step 2, a single cell is taken from each embryo for genetic analysis before implantation. This allows for the detection of genetic disorders like cystic fibrosis. Parents can then choose to only implant embryos without the defective alleles. Let's discuss some arguments for and against embryo screening. First of all, it prevents suffering. Embryo screening reduces the number of children born with serious genetic disorders and associated suffering. There are also economic benefits. It decreases healthcare costs by reducing the need for long-term medical care and treatment for genetic conditions. Additionally, there are large levels of regulation. Current laws control the use of this technology to prevent misuse, such as selecting for non-medical traits. Now let's discuss some arguments against. Firstly, there are ethical concerns. Some people believe that embryo screening suggests that people with genetic disorders are less valuable, which could increase societal prejudice. It can also be thought of as a slippery slope. This might lead to societal pressure to select embryos based on non-medical traits, like physical appearance or intelligence, leading to ethical and moral concerns. Additionally, there are high costs associated. Embryo screening is an expensive procedure, making it inaccessible to many people. This raises concerns about equity and fairness in healthcare. Finally, it can lead to incredibly difficult decisions. Finding a genetic disorder in an embryo already in the womb may lead to tough decisions, including the possibility of pregnancy termination. In today's lesson, we focused on inherited disorders, specifically cystic fibrosis and polydactyly. We learned that cystic fibrosis is caused by a recessive allele, leading to thick mucus affecting the respiratory and digestive systems, while polydactyly, caused by a dominant allele, results in extra fingers or toes. We also discussed the ethical, social, and economic aspects of embryo screening. Economically, it can reduce healthcare costs. Socially, it influences perceptions of genetic conditions. And ethically, it raises questions about the value of life and potential genetic discrimination. By understanding these disorders and the implications of screening, students can critically evaluate future medical technologies while considering ethical principles.